So we are starting the second half of the day with a little delay, but almost back on schedule. And it's a great pleasure. Our next speaker is Mark Schnitzer. He also second time visited us, and it was great because kind of it's getting a little bit more like in direction of discussions and developing things rather than having just meeting when people come, talk, and disappear and never go back to anything. So Mark is a HHMI investigator and professor in Stanford, and he is co-director of CNC Center. And do you guess what CNC means? Anybody can guess? CNC?
Center for Neural Computation.
Crack the neural code. I really like the title of the center. I'm wondering who invented it, and is it you, Mark? Anyway, so it's a great pleasure. So I think Mark doing most fantastic recordings of the brain, and if you ever will have kind of whole picture of the brain and what it's doing during sleep and wake, it would be coming from techniques like Mark developed in his lab. So. And it's a pleasure, and I'm looking forward for a very interesting talk. So please.
All right, thank you so much, Maxime. Can you hear me in the back okay? Yeah. All right, great. So thanks to Terry and to Maxime for the invitation. It's great to be back here. I really like this winter school. It's got a terrific tenor and atmosphere of sort of casual and yet very serious intellectually, so it's a lot of fun to be back and to see friends and people I haven't seen in a while. So what I want to convince you of today is that the voltage imaging techniques, I think, are really ready to start addressing the kind of challenges and questions we've seen in the talk so far today. And so that, if you take nothing else away from the talk, that's the message that I'd like to deliver. Now here's a slightly more detailed summary of what I'm going to talk about and what I hope you'll take away from the talk. So I'm going to start off, just for those of you that haven't seen this before, by talking about the FRET ops and genetically encoded voltage sensors, and I'm going to briefly review how they sense and report membrane voltage. And I'm going to tell you about how we have, at the moment, with collaborators, developed 4 different varieties of these FRET ops and indicators, which means in principle we can track up to 4 neuron classes at once. So far our record is 3 classes at once, but I don't think 4 is that far away in the future. And then I'm going to first describe how we've been using these indicators to look at spiking in genetically encoded neuron types. And I'll show you one little snippet of a story in which we were able to use voltage imaging to uncover a new form of memory interaction between shorter-term memories and longer-term memories. And after that, I will describe a method we call tempo for tracking voltage waves and oscillations in genetically targeted cell types. And so this is a population method. We're looking at aggregates of cells. Some people like to think of this as an optical version of LFPs. It's actually not, because we're looking directly at transmembrane voltages. We're not sensing extracellular potentials. So for example, there's no volume conduction artifacts and so forth. So I'm going to introduce you to the tempo approach, and then I'm going to describe work on a new fiber optic tempo system that we have, a photometry system, if you will, for freely moving animals. I'll tell you about some results with that. I'm going to describe a new tempo mesoscope that we have for direct visualization of brain waves. The little teaser you saw in the beginning on the first slide was from this system. And with both systems, we're able to look at coupling between distinct brain oscillations. This came up a little bit in the morning. I'll show you how we can directly visualize now cross-frequency coupling with cell type specificity. The tempo mesoscope has allowed us to uncover new forms of hippocampal waves. This work was done with Yuri, and in particular, we've found traveling beta waves. So beta oscillations were appreciated in the hippocampus, but I don't think people had visualized their travel directions. And we've also found a new travel direction for the theta. So I'll talk about this as we get there. And then I'm going to show you our ability to now track visually evoked cortical 3 to 7 hertz oscillations and gamma waves in response to visual stimuli. So just sort of a preview of where we're going. So let's start now with the FRET opsins. So these are the main indicators that we've been developing over the years, and they combine in a single fused protein, a voltage-sensitive opsin family protein with a bright genetically encoded fluorophore. So this is an attempt in a way to get the best of both worlds. When you put them together, these two optical moieties serve as a FRET donor and an acceptor, yielding a high SNR optical reportive membrane voltage. And this makes use of the fact that the opsin family proteins generally have voltage-sensitive absorption coefficients, which was first uncovered and highlighted by Adam Cohen. And so what Yi-Yang Gong did when he was in our group is instead of using the opsin protein directly as a voltage indicator, he developed this FRET opsin approach, which, as I said, is an attempt to get a very bright genetically encoded fluorescent protein and marry it with the voltage-sensitive opsin. So, as I said, Cohen had noted that in his case, arcuate acts as a voltage sensor by altering its light, its ability to absorb light, and in particular, neural depolarization raises the absorption coefficient. And in this case, that means it's going to, for our case, it means it's going to be a better FRET acceptor of the fluorescence energy coming from the donor. Now, this involves a protonation-deprotonation event, and so there's actually a migration of a hydrogen that changes the absorption of the opsin. And when we combine this with a bright genetically encoded fluorescent protein, for example, the bright fluor ASEM neon, then we can have a voltage-sensitive FRET reaction. And so just as a side note about the indicators, many FRET indicators that we use in biology actually involve a conformational change. This one really is not due to a conformational change per se, but it has to do with that change in the absorption spectrum. And the fact that the opsin family proteins typically have fairly wide absorption spectra means that you can actually couple them with different fluorophores of different colors. And I'm going to come back to that when we look at, for example, red indicators and green indicators, making use of the fact that if you, for example, look at this opsin spectrum, the absorption spectrum, there's room here for another FRET donor there. Now, Yang showed that these FRET opsin indicators can have a substantial fluorescence dynamic range of signaling. So this is some of the early ones we made, about 20% change in delta F over F, change in fluorescence per 100 millivolt, a change in the neuronal transmembrane potential. And the kinetics depend very much on the voltage-sensitive opsin that you've selected, and he identified the opsin from acetobulara as having really rapid kinetics that would be advantageous for reporting action potentials, 1 millisecond kinetics. And here you can see some step responses in different opsins. So you can see this particular one, this blue one, a variation of the acetobulara opsin, was sufficiently rapid to be able to detect the upswing of spikes, for example. And we can do this with modest amounts of illumination. So 15 milliwatts per millimeter squared of intensity, much lower, orders of magnitude lower than you would typically use with opsin family GEVIs if you're using the opsin directly. So here's an example from Yang's original data showing you simultaneously acquired traces, electrical and optical. You can see you're catching each and every spike very nicely. You can catch the waveforms, subthreshold things, and so forth. And of course, you can target this to specific neuron types of your selection. And I should say that other FRET opsin sensors work on the same principle that I've articulated, using the opsin as the FRET acceptor. A good example now is the chemigenetic Voltron sensors from Agilia Farm, using the same ACE opsin from acetobulara that Yi-Yang developed for his pure protein version. The Voltron, of course, uses a synthetic fluorophore as the FRET donor. Now, here's an example showing you that we can, with these these GEVIs with rapid kinetics, we can look at not only spiking, but also sub-millisecond aspects of neuronal dynamics. So this is from a fly neuron's movie Slow Down, and here you can see the back propagation of activity on the millisecond time scale. So we're able to really probe neuronal biophysics in a way that often has been difficult in the field. Moreover, we cannot just catch every spike, as I showed you in vitro. We can also do this in awake mice. So here, again, some of the original recordings that Yang did, showing you simultaneously recorded spikes done optically with ACE2NM neon green. And here you can see a micropipette with red dye snuggled up to the cell body for a very close extracellular recording. We're catching essentially every spike, and moreover, we can time the spikes in the optical records to within about 200 microsecond spike timing accuracy, as you can see from the histogram of errors there on the lower right. And we can do this in the fly brain. Here's, again, some simultaneous records, electrical and optical, using modest amounts of power. And I'm just going to zoom in very quickly to show you some of the spike waveforms. And the same kind of spike timing precision in the fly brain as well. Now, making use of the breadth of the absorption spectrum that I showed you in the opsons, one can imagine, and in fact can construct, Fred opsin indicators of different colors. So led by, at that time, research scientists and postdocs Madhu Kannan and Ganesh Vasan, who were working at Yale, and John Pierce with Vince Pirabon. We were collaborating with them. We came up with a red version and a green version. The red version was named Varnam, which I'm told means red in Sanskrit. And now there was the possibility of having two different neuron types labeled concurrently. You can look at spikes in both. And then most recently, the collaboration published a palette of 4 different Fred opsin indicators. And the way this works is that we have 2 green and 2 red indicators. And the voltage-sensitive polarity can either be positive or negative. And so this means that with a fairly standard dual-color fluorescence microscope, you can now look, in principle, up to 4 different neuron classes at once. And the idea is if you're looking at a particular neuron, first, you would ask whether the cell was fluorescing green or red, and whether the action potentials were upwards or downwards, and thereby disentangle which of the 4 cell classes you were looking at. Now here you can see on this voltage versus fluorescence change plot, the voltage sensitivities of the different indicators. So PACE stands for positive ACE, and you can see it has an opposite voltage sensitivity as the original ASM neon, or an improved version ASM neon 2. And Varnam also has a decrease in fluorescence with increased voltage due to the fact that you have better absorption in the opsin, whereas PACER is using the same configuration as PACE, but with a red fluorophore. So this is sort of showing you how we have both negative and positive polarity indicators of the 2 colors. So now I'm going to show you a snippet of sort of how we've been using this for new neuroscience. And the example that I'm going to give you is from flies. I'm not going to have time to go into all the details of this study, but just to give you a sense of sort of what we can do now with single spike level voltage imaging. So this is work by Chung Huang, and he now has his own group at Washington University in St. Louis. And he started off by trying to develop a chronic or long-term imaging preparation in the adult fly using laser microsurgery. And so in collaboration with Supriyo Sinha and Tom Baer, we built a laser microdissection apparatus based on a UV excimer 193 nanometer laser. So this is very similar to what would happen in LASIK if you went in for corneal surgery. And just a handful of nanosecond pulses from this laser, one can shave away the fly cuticle. This is illustrated here on the bottom. You can see before and after surgery. You can see the fluorescence of the fly mushroom body that is revealed by removing the exoskeleton. This can then be sealed with an optically transparent epoxy, and the transparency of this window then allows us to perform either long-term voltage imaging or long-term calcium imaging for periods of up to 50 days, which is more than enough to do novel experiments on long-term memory in the fly. And so here you can see some examples in which we've been able to come back in the examples shown here over periods of 10 days to individual dendrites or axonal boutons, and we can follow spiking or calcium if we're doing calcium imaging, again, for substantial periods of time. And we do this pretty routinely. And that points to the fact that the laser microdissection typically is far gentler than one can achieve with manual dissection in an adult fly. Now I'm going to show you what this looks like in a awake behaving fly. So here we have the fly on the ball prep. So you look at the lower left, you can see the fly is running there on the spherical treadmill. Meanwhile, Chung is acquiring high-speed optical video of the brain dynamics, so he's taking fluorescence videos at about 1 kilohertz, and in this case, he's actually tracking bilateral activity in a particular class of mushroom body output neuron and counting spikes in the green boxed area and the orange boxed area. And on the right part of this movie, you can see the spikes that are emerging in real time from these two bilateral neurons. Now, to sort of summarize for you what we did with this, we wanted to use the long-term imaging capability in conjunction with our ability to really catch each and every spike in the flies to probe memory dynamics in the fly mushroom body in a way that had not been done before. And so we focused in this particular study on the so-called PPL1 system of dopamine neurons. So this is a portion of the fly mushroom body that traditionally has been thought to be responsible for this learning and storage of aversive memories. And Chung systematically marched around this circuit. So there are parallel memory units that operate in this structure, in this circuit. And so we have actually 5 different dopamine neurons, different types of PPL1 neurons, that all have slightly different coding properties and encode slightly different things. And they serve essentially to provide teaching signals, for example, in an associative conditioning assay. And then we also looked, in addition to these 5 dopamine neurons, at 6 of these mushroom body output neurons that would serve more to guide the fly's behavior, avoidance approach, and so forth. And so because the fly circuitry is so well conserved and across different flies, and because we now have access to the connectome as well as the very sophisticated genetic tools, Chung was able to do voltage imaging experiments in 500 different flies, marching around between these 6 output neurons and the 5 dopamine neurons. And that's the kind of data set that I think most of us would agree would be prohibitive in mice. We're not going to do 500 mice in a study, but you can do it in flies. So I'm going to show you a little taste of what we found. Now, as I mentioned, these PPL1 dopamine neurons had been previously implicated in the encoding of aversive and the learning of aversive stimuli, such as punishments, for example, in associative conditioning assays. And this, in part, was due not just to behavioral and optogenetic experiments, but also due to the fact that in prior calcium imaging studies of these neurons, it was typically seen that it were reversive or punishment signals that drove these neurons to be excited. Now, with voltage imaging, we immediately got a different view of things because we were able to find that not only were these neurons excited by punishments, for example, electric shocks to the fly, but they were also inhibited by rewards, for example, the delivery of sucrose. And as you can imagine, if you know how calcium indicators work, it's this suppression of activity that is often not well reported in calcium imaging due to the rectification that occurs owing to the calcium indicators. But we could very nicely see the diminution in spike rates with these voltage indicators. And that bidirectionality, it turned out, proved to be critical as we went along in the study to reveal what these neurons were doing, and it proved to be critical for the computational model of the circuit that we later made. Now, it turns out that not only are these cells encoding aversive punishment signals or rewarding signals, such as the sucrose, in a bidirectional manner, that these dopamine neurons were also bidirectionally encoding the innate valences of different odors. So in a conditioning assay in flies, it's very common that you might pair, for example, an odor as a conditioned stimulus with another unconditioned stimulus, which might be innately rewarding or innately aversive. So it turned out that these neurons were not only encoding the valence of the US, in a sense, they were also encoding the valence of these stimuli, which are commonly used as the CS, the conditioned stimulus. And so here you can see examples. These are essentially spike rasters across the 5 different dopamine neurons that I mentioned to different odors, which are arranged from reversive to appetitive as we go down from the top to the bottom. You can see all these different neuron classes have slightly different responses, but you can see, perhaps with the exception of the neuron on the far right, they're essentially all bidirectional. And so then that raises the question, well, if we perform an associative conditioning task for these flies, we used an aversive memory paradigm, pairing CS plus odors with electric shocks. What might be the effect of the innate valence of the individual odors? So in other words, if you're pairing an attractive odor with an aversive unconditioned stimulus, might there be a difference in memory dynamics versus if you were pairing an aversive odor with an aversive unconditioned stimulus? And so we marched around this circuit with our voltage indicators, and we found that this learning paradigm induced distributed plasticity largely throughout the entire PPL1 M-bond circuit. And we found different forms of plasticity. So for example, in this particular pair of M-bonds, we found a short-lasting depression. This is going to have a short-lasting influence on the animal's behavior. In another one of these learning modules, we found a more long-lasting depression. And then, intriguingly, we also found a plasticity in some of the dopamine neurons themselves corresponding to a short-lasting learned valence. The point being is if through your learning paradigm you've altered spiking in one of the dopamine neurons, you essentially have altered the valence that is being conveyed about that cue. So we found plasticity not only in the output neurons but also in these teaching signals. We then took a look, as I said, at whether the pairing of this aversive electric shock with an odor of attractive innate valence or aversive innate valence would have any impact on the subsequent memory dynamics. And indeed it did. So we found basically valence-dependent memory effects. So if you look on the right, you can see that there is much more prolonged changes in spiking in response to a CS plus odor that's been paired with a shock. For a repulsive odor, we see sustained changes over 24 hours using our long-term imaging preparation, whereas with the attractive odor shown there on the left graph, you can see that there is a very substantial plasticity, but it recovers more readily, as if the system doesn't like to have conflicting information from the CS and the US. It doesn't imprint the memory as strongly. So when we looked at the different durations of memory storage in different aspects of the system, we came to the following hypothesis. So we took a look at the duration of plasticity in one of the outputs of the short-term memory module, and it corresponded quite well to the duration of plasticity in the input to one of these longer-term modules. And we knew that there was a feedback connection between the two. So our hypothesis then was that after the short-term memory has been formed, that you might have long-term memory storage owing to a disinhibition of this feedback from the output of the short-term memory module to the input to this dopamine neuron that is guiding plasticity in the long-term memory module. And so that became our hypothesis. I don't have time today to present to you all the evidence that we gathered for this, but just to say that Chung and Junjie Lo used knowledge of the fly connectome, which is now available with these imaging approaches, with computational modeling, with perturbative manipulations of the system, to gather a substantial amount of evidence that indeed that there's a gating of long-term memory storage by the activation of an existing short-term memory. And we believe this is a new form of interaction between short-term and long-term memory. It's different than the well-known consolidation of memory, which typically happens offline. This is a gating in the sense that it's not an offline interaction. It only happens when there's subsequent training, subsequent to the formation of the initial memory, that allows the long-term module now to encode a much more long-lasting memory trace. So I would encourage you to look at that if this is of interest. Now, we can do these kind of voltage imaging experiments with the FRET opsons, not only in awake behaving flies, but in awake behaving mice. So here's an example from the work of Simon Haziza in the lab. He's looking at two neuron types here in area V1, primary visual cortex of a running mouse, using just one of the green and one of the red sensors. And they were labeled by different projection targetings of different viruses. And you can see that here, in layer 2-3 of the cortex, we can nevertheless find interspersed different populations of pyramidal cells with different projections. So this is a new means now of disentangling how excitatory neurons with different projection profiles might differentially encode information. Now here's an example of Simon doing simultaneous voltage imaging with 3 neuron classes in area C, one of the hippocampus of a running mouse. So here he's used the 2 different green indicators to selectively label either entorhinal cortical projecting CA1 neurons or lateral septum projecting pyramidal cells, or using a Cree driver line, he's labeled with a red indicator the somatostatin interneurons. So this just shows you we can now begin to track multiple classes of neurons concurrently. And I want to also call out in this slide work that we did collaboratively with Adam Bowman when he was at Stanford. He's now here at Salk as a fellow. He was with Mark Kastovich at Stanford, and he and Chung looked at the fly with our Fred Opson indicators. And one of the interesting things about these indicators is they not only have a voltage-sensitive emission of fluorescence intensity, but also the fluorescence lifetime is changed in a voltage-sensitive manner, owing to the modulation of the FRET efficiency. And so what Adam and Chung showed in the flies is that by using a electro-optic wide-field fluorescence lifetime microscope that Adam built, they were able to get very high SNR reports of flies spiking in a way that often was superior to the intensity measurements, as you can see here from the blue traces, which are the lifetime reports, and the black traces, which are the traditional intensity reports. So the use of voltage indicators that have lifetime changes may be a very promising avenue forward for the field. Not all the indicators that we use necessarily have these substantial lifetime changes, but for the subset that do, this may be a very promising way forward. Now I'm going to shift gears a little bit and turn from the imaging of spikes to the imaging of electrical oscillations and waves. And these objects of study have obviously been known for a long time now, nearing 100 years. And one of the things that's been missing nonetheless is the ability to selectively image waves and oscillations in neuron types of an experimenter choice. And so a number of years ago, Jesse Marshall in the lab, working with Michael Lin, who's a protein engineer and makes voltage indicators as well at Stanford, Jesse developed the TEMPO concept, which stands for transmembrane electrical measurements performed optically. And it's a means, as I said earlier, of looking at population level subthreshold voltage activity. And here you can see some early examples in which Jesse is performing a TEMPO measurement targeted to pyramidal cells. At the same time, he's taking an LFP recording. And here you can see some of the nice concordance between the two different modalities. Now, to sort of briefly summarize for you before I get into a little bit more detail how TEMPO works, either in the fiber optic version or in the imaging version, it has 4 basic ingredients. So typically we would select individual neuron types, either with viruses, transgenic animals, or other means of selective labeling. We always use a reference fluorophore that is not voltage sensitive, and the purpose here is to be able to track hemodynamic, brain motion, or other artifacts that might reduce our ability to see voltage transients. Many of you may know very well historically that some of these other effects which are dependent on brain activity actually used to be used by the optical brain imaging community, such as intrinsic signals for detecting the activation of specific brain areas. Third, we always use a dual color fluorescence measurement apparatus to track our genetically encoded fluor and a reference fluor, or our 2 different voltage indicators and a reference fluor. And then we computationally unmix the contribution of the artifact signals from the records of neuronal voltage activity. Now, recently, Simon Haziza in the lab put together a fiber optic version of this measurement approach we call TEMPO 2.0, and it's about 10 times more sensitive, we believe, than any prior fiber photometry system used for voltage sensing. I don't have time today to go through all the details of the particular optical engineering advances, but just to summarize, there's a low noise illumination module. There's a module for breaking the decoherence of the laser light. We do phase sensitive detection with modulated light sources, and there's a low noise fluorescence sensing module. And then after the experiments, we computationally unmix, as I was saying, the effects of hemodynamic and other artifacts. Now, our approach to unmixing was developed by Vasily Kruzilin, who's here at the workshop. And the idea that he promoted was that our static fluorophore, in many cases it's a red fluorophore while we're doing voltage imaging in a green channel, is going to be impacted by a variety of different signatures, heartbeat, breathing, possibly even electric line noise, that have different frequency signatures, and that will all affect the red and the green fluorescence channels slightly differently. And so therefore, he came up with an approach to do a frequency-dependent unmixing of these artifacts between the 2 channels. So first, he will form a filter that will allow us subsequently to do a convolutional unmixing of these effects, and then he applies the filter after learning the optimum filter to the data. He does this in the frequency domain. And this is very important because, for example, if you just do a frequency independent unmixing, the extent to which your breathing artifact affects both channels may be different from the extent to which for example, your cardiac artifacts may affect both channels. So you want to be able to unmix in a frequency-dependent way. They may have different phase shifts across the different frequencies. So here's just an example of what one of these filters looks like in the time domain on the left and in the frequency domain on the right. And so you can see that it's able to account for these different artifacts in a differential way that takes into account the fact they really have different sources and different impact on the two fluorescence channels. So we apply this approach in all of our data. So here's some examples now from the fiber optic approach. So to start off, I'm showing you data from ketamine xylazine anesthetized mouse. And we can see both delta and gamma oscillations concurrently. And when we look at a spectrogram, as shown there on the lower left, we can actually see there's two different forms of the gamma, a high gamma and a low gamma, with slightly different phase shifts relative to the up-down of the delta. Now, in awake behaving animals and in work that we did with Yuri, Simon was able to demonstrate, as you might expect, the locomotor dependence of the hippocampal theta and beta rhythms using tempo that was targeted to CA1 hippocampal parvalbumin interneurons. And so if you look at the very top line there, that's the locomotor speed of the animal, and you can see that when it runs, we see enhanced power in the LFP, in theta and also beta, and particularly we see nice elevation and coherence between the tempo signal and the LFP in these two bands, theta and beta, when the animal runs. Now, we were able to do this in a cell type dependent manner using a pair of voltage indicators of different colors. So here, Simon has targeted a red voltage indicator, the Varnum, to CA1 pyramidal cells, and the green indicator to the parvalbumin interneurons. And the top four plots are for when the animal is at rest, the bottom four plots when the animals are running. You can see a shift in those low frequency bands to slightly higher frequencies, i.e. like the increase in the theta when the animal runs. But you can also see a laminar-dependent increase here in the beta. So, for example, what we found in both neuron types, but particularly in the parvalbumin cells, there's a clear enhancement in the beta frequencies in stratum radiatum. And that is determined using a silicon electrode implanted into the, with multiple contacts implanted into the contralateral hippocampus. Now, there's been a lot of talk about ripples here, and I just want to say that we can use this approach to try to dissect the contributions of different neuron types to the ripple. So here you can see after isolating the pyramidal cell layer with a silicon probe, we can then perform the tempo in the two different neuron types, and you can see the differential activity of the PV interneurons in the pyramidal cells and this sharp decline in PV activity that occurs immediately right after the ripple. Now, we've also been able to generalize these fiber optic approaches to tempo imaging with the new mesoscope. This was an instrument built by Radek Krapkiewicz, again, targeting specific neuron types. So here you can see the example that I showed you in the first slide, traveling cortical delta waves. You're visualizing this through a 7 millimeter diameter cranial window in layer 2-3 pyramidal cells. And when we looked a little bit more carefully at these waves in ketamine xylazine anesthetized animals, we found examples of cross-frequency coupling, as has been documented, with the gamma now embedded in the depolarization phase of the delta wave. At the bottom there, you can see two very nice examples. And here you can see some snapshots of these waves traveling across the cortex. And what I want to point out here in these next two slides is that although cross-frequency coupling has been appreciated, what I'm going to show you is that these two waves of different frequencies are not traveling in the same direction. They're actually traveling orthogonally. So here at the top, you'll see two examples of delta waves going anterior to posterior with relatively low speeds, 12 to 15 millimeters a second. Here's some histograms of the speed and the direction. And then when we look at these cross-frequency coupled gamma waves, here again are two examples. You can see them traveling in a very different direction, traveling lateral to medial with much higher speed. So not only are they coupled, but they're headed in orthogonal directions. So that's, I think, a very interesting phenomenon. we can now directly visualize. Now, I mentioned at the beginning that we've been able to uncover some new traveling directions for hippocampal waves. We can do this in a cell type specific manner. So we looked at hippocampal beta and theta waves in an animal that's head fixed under the tempo mesoscope. And we found that both beta and theta waves in hippocampus are traveling waves that can propagate in two different directions. So here at the top, you'll see examples of individual beta waves. And these arrows here on the far right of each set of snapshots shows you the direction of travel. So we have a pair of orthogonal travel directions for these beta waves. You can see that they can go septal to temporal or C in the C3 to C1 direction. And they're moving pretty quickly at about 220 millimeters per second. And we then looked at theta waves, and we found that these can also propagate in an approximately bidirectional manner, and I believe only one direction has been reported previously. And so here you can see that at least in PV cells, we find that you can have forward propagation and also backwards propagation along the C3, C1 axis. And you can see how much slower these waves are. But in this case, the speed seems to be dependent on the cell type that we're looking at. So in this case, we used a PV Cree driver line to target parvalbumin cells, but then we used projection targeting to look at lateral septum projecting excitatory cells. And we only found one direction of propagation in this population, suggesting that perhaps they don't participate in the other. Now, as I mentioned, we also looked at sensory evoked waves. We looked at 5 different neuron types in the visual cortex after presentation of a visual grating. And here, what you can see as the mouse views the moving grating, that there typically is an elevation in gamma power during visual stimulus presentation, followed by a 3 to 7 hertz alpha-like wave at the visual stimulus offset. I'm going to show you what these waves look like. So this is a higher speed video taken from V1 on the left, and they're very nicely at the end of the movie. I think you can see a traveling gamma. So it'll loop around so you can see it again. And you're seeing the much slower wave there on the top version. So we can begin to directly visualize these waves now. And so I showed you 2 cell types there, and here we have 3 more cell types across layer 2, 3, layer 4, and then layer 5 pyramidal cells at the bottom. So we see the 3 to 7 hertz and the gamma waves in all these cell types, but with slightly different spatial temporal attributes. Now, with the dual Gevi tempo, we were able to look at the relative contributions of the PV cells in the layer 2, 3 pyramids to these visually evoked 3 to 7 hertz waves. And we could do this with assigning the green and the red indicators to the interneurons and the pyramidal cells, or we could flip them to make sure that any phase lag was bona fide and was not due to the indicators themselves. And so what we found was that there was a subtle, on the order of 2 millisecond offset between the pyramidal cells and the PV cells. And this was the result regardless of when we swapped the indicators. So now I'm trying to show you that we can really begin to look at some of the subtleties, the cell type contributions to waves of this type. So to sum up, this is just what I said at the beginning, going from the voltage indicators to the newfound ability to record from spikes in different neuron classes, to be able to directly look at these different forms of waves and oscillations. And I tried to thank members of our voltage imaging team as we went along, but here are some additional photographs, and many thanks to the collaborators. So thank you.
Hi, yeah, really interesting talk. It's exciting to see how quickly this is developing. One of the things I was wondering was, so with a lot of the calcium imaging field with these mesoscopic kind of like large field calcium imaging experiments, there's been a lot of work people looking into these specific different kind of like spatial temporal patterns of activity and how you can kind of cluster them into a relatively small number of patterns in a way. Is there any kind of interest in your group or others that you know of to use these kind of fast, high temporal resolutions to do similar kind of thing where you're trying to look at maybe representation?
So you're talking about things like non-negative matrix?
Yeah, and looking to see if there's kind of specific like states or patterns of activity, spatial temporal activity across the cortex.
I mean, you obviously can run those algorithms.
Yeah.
I mean, I think one of the things that I hope that these movies convey is there's a lot more richness in these voltage movies than there is in the calcium movies. So, when I look at the calcium movies, I mean, I like those too. You tend to see these blobs coming up and they go down, right? So you're sort of looking at brain areas of activation. But here, we're looking at traveling waves. And so, NMF is not necessarily the best way to doing that, right? We're really working at the level of single trial events, and I didn't talk too much about this, but, because I was mostly showing you fairly stereotyped directions, but some of these wave types are not stereotyped in their directions. So for example, those gamma waves that I showed you that were visually evoked, they actually have, if you look at the statistical distribution of those waves across different trials, it's actually quite isotropic. So meaning that on a given trial, you may find a highly directional wave, but if you look across all the trials upon which you might, for example, consider an NMF analysis, now you're looking at waves across all different directions. So I worry a little bit that if you apply some of these blobology analytics, you may sweep under the rug a lot of trial-to-trial variability in speed, direction, phase shift, and so forth. So,
Very exciting.
yeah,
Thank you.
I think that would be the concern.
Thank you for the amazing talk. I've got a naive question which actually follows up what you were just saying. Do you ever see these waves actually not being, you present them as if they were planar waves, and people always talk about that, but are they spiral waves really? Have you looked for that?
Well, Terry has reported spiral waves, right?
Yes.
I would say we have not really seen spirals yet, but I think we certainly could.
I mean, when I was looking at the patterns that you were showing in the movies, you can see bursts that sort of appear to have some sort of angular momentum. I don't know if you...
Oh, in the movies that I showed you?
Yes. I mean, I was looking at it because I've generated similar types of movies in other contexts.
Yeah,
there's a theoretical prejudice for why you might want to have...
Yeah, it certainly could be. I mean, I think, we would want to do it systematically and,
Yes.
but no, I mean, we're not reporting any spiral waves yet, but I think we certainly have the sensitivity to do so, yeah.
Yep, thanks.
Yeah, just to follow up, yeah, we found that in sleep, where the cortex goes in these low frequency amplitude modes, that we see spindle waves that are systematically traveling around the entire global cortex in a spiral way, circular. And by the way, it also reverses direction, and sometimes you get expansion. So it's just the way you described it, is that it's really heterogeneous. Okay, so these aren't really waves, they're packets. That is to say, if you look at the spindle, as I was saying, it lasts about 2 seconds. It's 10 to 14 hertz. That you showed gamma, and it looked like a packet that lasted just a few hundred milliseconds. And can you say something about how the size of the packet varies with the frequency?
I'm not sure we've looked at that in a systematic way. I mean, I think you could see from the movies of the delta that I was, showing you, which is an anesthetized case, right? It's really quite substantial, lower speed, longer wavelengths. But, if you're asking, have we, for example, tried to put together a dispersion relationship or something, like that kind of analysis? So maybe you're asking me, have we, done a fine-grained analysis of the different gammas? And looked at longevity, looked at frequency. We haven't done that kind of systematic disentangling.
Right.
It's a great question, though.
Yeah. The other thing is that these are actually broadband in the sense that it's not a single frequency like 40 hertz. It's like 30 to 80, which means that the period varies from cycle to cycle, and that's also often ignored. It's really much more complex.
Yeah. Yeah. No, these are wonderful questions. I mean, I think your question really highlights, I think, the virtues of having single trial resolution. So a lot of the work that's been done, has really done a lot of trial averaging, and obviously some of these waves are locked to the stimulus presentation and some are not, right? And so if you're looking at a wave that's not locked, you're going to average it out in some sense, and so I think these kind of questions that you're posing will very much take advantage of the fact that we now have single trial resolution, in both the fiber optic and the imaging apparatus. So I don't have specific answers for you today, but I would say, the field is ready to address these kind of questions.
This is really wonderful. It's really exciting to see this happen because it's something that especially the trial averaging is a tradition in neuroscience going way back. And now we're beginning to realize that that was really averaging over a lot of very important detail.
Hi. Hi, thanks for the talk. Mine is a very simple question. You showed us like some videos of the imaging in fruit flies, and then you had like this plot with the left hemisphere and right hemisphere. And usually when we see these calcium imaging things, the resolution is like at single cell level, and we can see like specific cells there.
Yeah.
But in that video you showed, it seemed that it was more zoomed out. So but you did show from the plots you showed.
Yeah, so just to clarify a little bit, so the optical magnification is not the same as the single cell resolution, right? Because in flies, we're typically using some very specific Gal-4 lines to target the single cell types. Does that answer your question?
Yeah, that's exactly what I want to clarify. So, but you did have the single. It seemed that you had like the voltage of a single cell there in your slide. That was I was wondering if that came from electrophysiology or is it from the image?
This?
Yeah, this.
No, this is directly from the camera.
Yeah, but that resolution, is that a single cell that we see there in the video?
Yes, because of the virtue of the targeting. So what Chung is doing here, he's literally tracking the fluorescence in those boxes.
And the box is just one cell there? Is that one mushroom body neuron?
On each side, right, because of the targeting.
Oh, okay.
Right?
I thought it was more
the
zoomed
point
out.
is the point is that the specificity is provided by the genetics. It's provided by the genetics.
And you can see both right and left hemisphere in just one image there? Is it that small?
I mean, yeah. I mean, look at the scale bar, 20 microns. See the scale bar is pretty small.
Okay, cool. Yeah, I wanted to clarify. Thanks.
Yeah, you're very welcome.
Hey, so we have these voltage indicators. We're putting a lot of them in because you were able to see these waves kind of traveling within the neuron itself. Are we worried about molecular crowding? Are we worried that things are different than they would be like without adding these proteins in, or is there just enough room at the membrane that you're not concerned about things being different?
So I think you're asking if the voltage indicators potentially have some deleterious effects of their own. Is that the question? Yeah. So, first of all, so there's a variety of things to be concerned about potentially. So, the fluorescence generation process is typically associated with photobleaching and photodamage, which are generally thought to have to do with the driving of the fluorophore into a triplet excited state, which is more photochemically reactive. Okay, so that's potentially a concern. You have to sort of watch the health of your neurons, which will track in some way the bleaching. So you can have sort of an easy way of sort of following that, and you have to be careful not to provide too much light. There are also other issues that can come up with voltage indicators. So for example, you're stuffing these proteins into the cell membrane, you may be changing the capacitance, and you can sort of watch that. It's going to be very much dependent on the expression level, so you don't want to overexpress them, so you don't want to make sure you're not changing your spike waveforms and things like that. So there are things to be concerned about, and I think my perspective would be that these are manageable, but that you should be on the lookout.
Is there time for one more question, or?
A quick one.
Okay, well, so two quick ones, but really quick. One is, and I'll ask them quick and sit down. One is, when you showed us the whole brain, would you be able to do that with action potentials as well? And the second one is, when I look at this, and because I've done this sort of thing, but with voltage, I wonder what's a signal to noise? How much of the membrane potential are you explaining with the traveling wave?
Yeah. Well, the first question is sort of easier because it's a hardware issue, and the simple answer is I don't think the cameras are quite there yet, but I suspect it will come. Now there's always complications, with regard to depth and cell type and targeting. But if you're just asking a pure instrumentation question, then I think we will eventually get there, but we're not quite there. In terms of your second question, there's quite a bit of complexity still in terms of where this tempo signal is coming from in the following sense. So even though it doesn't have the complexity of being an extracellular potential, so we don't have to worry, for example, about conduction artifact, in principle, the different cell types will have different proportions of membrane area devoted to cell body or axon or dendrite of different compartments and so forth, right? And moreover, to even make it more complicated, your ability to access these different compartments may depend on the geometry of the neuron, right? So for example, the basal dendrites in the hippocampus are more dorsal, right? Whereas in the cortex, they're more ventral. And so, we're still working through, those issues of sort of, how best do we interpret the signal with different neuron types, with different targeting strategies, different geometries. We can perhaps use targeting, target it to the dendrites only, target it to the axons. So, but we're going to have to work